Okay, welcome back everyone. Thanks for joining us again for the multimodal workshop this afternoon. It is an absolute pleasure to introduce our next speaker, Danielle Bassett. Please give her a warm welcome.
Thank you. Thank you. Thanks to the organizers for inviting me to this workshop. I'm really excited to be here. I think that this topic is extremely exciting, very timely, and important as we look forward in the direction of the field. So I titled the talk, The Utility of Multimodal Neuroimaging in Building Network Models. And when I was thinking through, try what? On the slide. Ooh, okay, thank you. When I was thinking about what would make sense to talk about today to this group, I was trying to think and consolidate what I think the utility is of multimodal neuroimaging. And here are three different points that I think are relevant. Obviously, I'm sure that over the last few days you've heard about many others. The first is to explain observations in a modality late in the causal chain from observations in a different modality earlier in the causal chain. And that is going to depend on whether or not we can actually determine the state, the causal link inside of different modalities, but let's just put it up there as a potential. Number two is to build generative models that can bolster inferences from sparsely sampled data. And then number three is to inform mechanistic models. And these are the three topics that I'm going to go through today. I decided to focus on three studies, one of which it was just published this year, a second one which is just on arXiv as a preprint, and then a third that was just accepted in principle. So these are newer work that I think may be relevant and of interest to this community. Okay, so the first one is where we focus on the very first topic. I'm going to begin by talking a little bit about high-resolution human neuroimaging using electrocorticography. So as we know, electrocorticography provides a very beautiful direct measure of neural activity in humans, but it's been challenging for many years now to understand the patterns of functional connectivity that are present in ECOG at the whole brain level. And largely that's because there are three key challenges. One is that the measurement modality is invasive, which means that we cannot actually acquire this in most humans. It is usually only used for localizing seizure foci in medically refractory epilepsy. Number two is that there's often incomplete cortical coverage, and largely that's because of patient safety. And then number three, there is a variability in electrode placement across individuals, and so we do not get a whole brain measurement of activity or functional connectivity across the entire system. And that variability in electrode placement is again because surgeons are guided by what is clinically relevant for that particular person. So these three challenges, however, I think are potentially opportunities for multimodal imaging. And so in this study, what we decided to do was to collate information, electrocorticography information, across many different individuals with the aim of synthesizing a whole brain, interregional, and band-limited ECOG network from this multimodal or multi-subject database. We'd like to describe whole brain connectivity even though any particular individual is sampled in a sparse way. And then our second aim was to explain electrocorticography network organization as reflecting communication along white matter pathways, potentially being modulated by interregional Euclidean distance between different areas, and being explained to some degree by correlated gene expression. And so I'm going to use these little icons here throughout the next little bit. So here is the functional connectivity of ECOG. We're going to ask whether it can be explained by patterns of communication across white matter, patterns of gene co-expression, and then Euclidean distance between two different regions. Okay, but I'm going to start with aim one first, and that is to synthesize whole brain ECOG networks. To address this aim, we used publicly available data from the DARPA RAM project. At the time that we downloaded the data, there were 86 patients in the cohort. There are now over 300. And so this is a really beautiful public resource that anyone can use for similar types of questions. We extracted resting state periods in between sessions of a free recall task because we were interested in endogenous and intrinsic activity in the brain. And then third, we estimated functional connectivity using a simple simple correlation coefficient, although there are obviously other ways that we could do that in a more sophisticated way. What I'm showing you here is just for a single individual and for a particular trial in between the free recall task sessions, we have sparse sampling, but from every electrode to every other electrode, we can calculate the correlation coefficient, and then we can do that for every single trial. We can collapse all of these down into a single inter-electrode graph by averaging these together across the different trials for a single subject. But then we're faced with the challenge of collating the data across different subjects that have electrodes in different areas of the brain. And so to do that, we identified where, using MRI, we identified where the electrodes were on the cortical surface. And then we used a parcellation, a whole brain parcellation, to identify the similarity or the distance from every electrode to the center of mass of an ROI, so a region of interest that we identified using the Schaefer parcellation. And what you're seeing here is just that we can go from the inter-electrode graph then to an inter-regional graph, where these regions are defined from the cortical surface. And every element in here now is going to be a collation of information across all of the electrodes in each region that were placed for different subjects, and they are weighted by how close each of those electrodes was to the center of mass of the region. And so that basically allows us to go from the single subject level using inter-electrode graphs to the across subject level using interregional graphs. Here's just a map of the location of all of the electrodes for the entire group so that you can see that we do get very broad coverage, particularly on the lateral surfaces of the cortex. There is also some coverage in the medial wall also, but not quite as much as on the lateral surfaces. And then here you see the 86 individuals, and then here across those 86 individuals, once they're in region-to-region space, then we can take the average of those matrices to construct a whole brain correlation matrix for the entire group. We can do it for single subjects, but then we can also collapse over that information to study the entire group. So this is the overall process of creating that network. And our first question was, what is its architecture, and does it share some similar principles to what we see in other types of data, and specifically in resting state fMRI data? So I'm just going to show you one piece of information from that analysis, and that is the architecture of this process when we just look at the band of 1 to 4 hertz. We did the same procedure across many different bands from low delta up through high gamma, but I'm just going to show you the 1 to 4 hertz band here because that was the one where the pattern of functional connectivity was very similar to what we found in the resting state functional MRI. And so here what you are seeing are different brain regions along the y-axis, and along the x-axis are iterations of a modularity maximization algorithm, which is an algorithm it's basically a clustering algorithm to identify groups of regions that are strongly functionally connected to one another. And what you're seeing here is an index vector, which says across all of those iterations, these are the regions that are located in one community. And we apply a clustering algorithm to that whole set to identify each of these different clusters. On the bottom here, what I'm showing you is just if you take all of those regions that are highlighted in green here with some probability, so averaging across the x-axis, then you come up with this surface visualization. And what you can see is that these regions in this cluster from 1 to 4 hertz whole brain ECOG networks looks very similar to the dorsal attention system that we would have seen in bold fMRI data. Here is a similar cluster here in red that when you map it onto the brain looks very reminiscent of the default mode system. Here's one that you see that's very similar to the limbic system, the salience, the frontal parietal cognitive control, the visual system, and then the somatomotor system. So what I think is interesting about this is that by extrapolating patterns of single subject undersampled data to a group level, fully sampled correlation matrix, we obtain architectures that are very reminiscent of what we see in resting state bold fMRI data. I will say that this pattern of modular structure that you see here is not as strong in the higher frequency bands. So it is to some degree present across several of the frequency bands, but it's really the strongest in this low frequency 1 to 4 hertz band. So after performing that analysis, the next thing that we wanted to do was to move on to aim 2, which is to explain that pattern of network organization as reflecting different components that we may be able to identify or probe using very different kinds of imaging and information modalities. So our question was, if we take that whole brain functional connectivity network from all of the electrocorticography data, either at the low frequency band or the high frequency band, I'm actually going to show you all of them in this next set, we can ask the question of whether we can explain that using patterns of communication, patterns of distance between regions, and then patterns of gene co-expression. I'm just going to explain to you what each of these pieces is, but what you can basically see from this architecture is that this is a linear combination. So each of these matrices is multiplied by a beta weight. So this is going to be the beta for communication, this is going to be the beta for distance, here's the beta for expression, and then we'll have a constant term at the end. And in asking whether this can be predicted, we're basically tuning these beta values and asking, is there a set of beta values which, when multiplied by these 3 matrices, gives us the functional connectivity of the electrocorticography data. Okay, so what are each of these? The simplest one is the Euclidean distance. So here we take the 400 regions from the Schaefer atlas and we estimate the distances, physical distances between them. So that's just from center of mass of one region to center of mass of the other region. And then probably the other next simplest is this one, search information. So this is actually from diffusion weighted MRI from a large healthy cohort. And we perform deterministic tractography on that diffusion imaging data and then calculate the search information, which is basically a measure of the hiddenness of shortest anatomical paths from region I to region J. And you might ask, why do we use search information? The reason is that there was this very beautiful paper from Joaquin Goni a couple of years ago in the Proceedings of the National Academy of the Sciences that showed that the search information estimated from diffusion imaging data is one of the strongest predictors we have so far of resting state fold functional connectivity. So it is the best predictor from structure. So we're using that as our measurement of communication, the Euclidean distance, and then the last bit was the gene co-expression. So here we used gene expression data from the Allen Brain Institute, and we used 2 versions of it. Again, we assigned the different probes to specific regions from the atlas, and then we estimated the correlation between the levels of expression. We either used all of the genes, and for that I'm just going to call the matrix G, or we used the set of genes that was optimized for predicting resting state functional connectivity that was published in Jonas Ricciardi's paper in Science from 2015. And I'm going to call that G-optimized or G-opt. So this is either all of the genes from that data set or the optimized set published by Ricciardi. Okay, so that is the overall setup of the optimization. So then the next question is, can we take each of these pieces, which are going to be consistent for all individuals, right? It's one pattern of gene expression, it's one pattern of Euclidean distance from the atlas, and it's one pattern of search information from a separate healthy group of diffusion imaging data. So we take all of those and we ask, can we tune these beta values so that we can optimally predict the pattern of electrocorticography functional connectivity? Then we compare it to the observed functional connectivity and ask what the similarities and differences are. So just a quick visualization before we get into statistics. Here what I'm showing you is the pattern of observed functional connectivity on the left-hand side, and then the predicted functional connectivity on the right-hand side for the 1 to 4 hertz band and for the 140 to 165 hertz band. So very low frequency and very high frequency. And just by eye, what you can see is that there are some very nice correspondences between the observed data and the predicted data, both at the low frequency and at the high frequency. But what we'd like to do is to actually quantify those and also to understand which of the matrices, so gene co-expression, search information, or Euclidean distance, are mostly driving these effects, if there is one. All right, so here, let me just walk you through, there's a lot of data here. Here's the correlation on the y-axis between the observed functional connectivity and the predicted functional connectivity. Here is the 1 to 4 hertz bands all the way up through to the 140 to 165 hertz bands. And then in colors, you are seeing which piece did we include inside of the model. So first you have the just single pieces alone. So if we only include the distance matrix, that's the dark blue that comes first. If we only include the gene expression matrix, that's the lighter blue. If we only include the optimized gene expression, so the Ricciardi set, that's in this green color. If we only include search information, it's in this olive color. And then here you see pairs, so the optimized gene expression and search information, or the same with distance, or search information and distance. And then the last one, this dark red, is the combination of all three. So what do you see on average? Well, there are a couple take-homes from this graph. The first one is that in general, the patterns of functional connectivity in a low frequency band are less well explained by our model than the patterns of functional connectivity in the high frequency band. So on average, these values are lower, more around 0.4, and on average, these values are higher, closer to about 0.6. So that's the first observation that we can make. The second observation that we can make is that if you only study the models that include one type of information as the predictor, then Euclidean distance seems to do the best. So Euclidean distance is this dark blue. Notice how high that is consistently across all of these graphs. So the Euclidean distance is one of the best predictors if you have to use one alone. Gene expression is the worst. So that's the second blue. Notice how small that second blue bar is consistently across all of these different frequency bands. And then the third observation that we can make is that the combined model that includes all three outperforms the single and pairwise models. And in fact, it's the best model for all of these frequency bands except for the 1 to 4 hertz, where the yellow is the best. And the yellow is the optimized gene co-expression and the search information. Now, how would we want to use this sort of information? I think one of the ways that we might want to use it is to predict single subject electrocorticography functional connectivity in an unseen patient. So to address whether that was possible and to what degree we could do that well versus not do that well, we took all of the subjects and we left one out and calculated the functional connectivity of n-1, and then asked whether we could predict that and everyone except one, and then using those specific parameters, we estimated what the functional connectivity would be from the held out subject. So what I'm showing you here on the right-hand side is the correlation magnitude between the observed and predicted electrocorticography functional connectivity, again for all different frequency bands, and every data point here is for leaving one subject out. So I think that there are two observations that we can make from this data, which is that in general, we seem to be doing better at predicting the left out subject as we go to higher frequency bands, and we do significantly worse in the lower frequency bands. That overall trend is consistent with what we saw when we just used the entire data set here, right? But the second observation that I think is important is that there's a large amount of individual variability. So some people, we can predict their functional connectivity very well from the group information, and then others we can predict very poorly from the group information. At the moment, we don't understand what it is about a single subject that allows us to predict really well or to predict very poorly. So that's, I think, an opportunity for future work to explain this variation and to understand who is it for whom we can predict their functional connectivity quite well or not quite well. And I think this becomes important when we think about the fact that it's possible that it would be useful to be able to predict functional connectivity outside of the currently sampled zone. There are cases where, for epilepsy subjects, the neurosurgeon has already gone in and put in the grids, and then they realize from the time series during a seizure that they don't think that they've identified the seizure focus with high probability, and therefore might want to add in more grids, and that's not something that they would want to do for the safety of the patient. But if we were able to predict some of the information that was unseen, that may help them in determining the seizure focus. Okay, So in summary, for this piece, what we wanted to do was to synthesize whole brain interregional and band-limited electrocorticography networks from multi-subject data, which we did. And then after constructing that functional connectivity for each subject and then across all subjects for the group, we asked to what degree we could explain that pattern using communication patterns over white matter, gene co-expression, and then distance between different regions. And we found that combining all three together, we are able to have a not fully accurate, but a somewhat accurate prediction of the average functional connectivity. And we are also able to show that we can predict some single subject ECOG networks, although we are poorer at others. And we're interested in understanding those individual differences in future work. So I think there's some opportunities here for this kind of modeling. When we're asking about multimodal data, could we take this approach and use it to ask different sorts of questions? One question that I'm very interested in is to ask, is it possible that distinct brain states are associated with different models of this type? So if we took the functional connectivity data and we looked specifically during low arousal states or high arousal states, would we find that different pieces of this equation become more important? And particularly in high arousal states, I would predict that actually direct routes along white matter structure are going to be very important. And in low arousal states, those direct connections may be less important, and we may see a lot more happening along indirect pathways, similar to what the search information is able to provide. We have some preliminary data supporting this notion, but I think it's we need to do a bit more to consolidate that evidence. Another opportunity using this same kind of modeling approach is to ask whether we can understand the impact of expression patterns that are specific to disease-related genetic loci on observed functional connectivity patterns. So in particular, if we were to use, for example, the 108 schizophrenia genetic loci that have been published previously, would we find that a different pattern of functional connectivity is predicted from them versus the optimized gene set that was identified from healthy individuals? And if so, would we be able to combine the predictions of the disease-related genes and their impact on functional connectivity and the non-disease-related genes and their impact on functional connectivity to better understand what we actually observe in the patient or in the patient cohort? And then lastly, and this is the one that I think has very important relevance to neurologists and neurosurgeons studying medically refractory epilepsy, is the question of whether we can use this multimodal, multi-subject model that we are building from a large cohort of individuals that have previously been imaged to understand and predict unsampled electrocorticography data from a new patient. So I think that it's possible that we will always have undersampled ECOG data. That's going, in fact, the undersampling issue is increasing with time because neurosurgeons are moving from grids and strips to stereo, which provides less cortical coverage, although it does allow some additional white matter coverage. So the undersampling will remain a problem, but if we can determine what it is that we would expect from a whole group of individuals that have undersampled bits, but we combine it together to understand the global structure, then perhaps we can identify variations here from that sort of prior. And I'm hopeful that that could help us in the future to determine if we've captured the whole epilepsy network or if we need to go in with additional samples. Okay, so that brings me to the second part of the talk. In the first bit, what I wanted to go through was understanding whether we can use multimodal neuroimaging to explain observations in a modality late in the causal chain from one early in the causal chain. And I think it's an interesting question of what is late and what is early. One could suggest that structural connectivity is something earlier in a causal chain than functional connectivity, in the sense that functional connectivity is something that occurs on top of structure and is constrained by structure. But I think that there is more to do to better understand what that ordering of modalities should be. And then number two, to build generative models that can bolster inferences from sparsely sampled patient data. I want to move into the last bit to inform mechanistic models, but before I do, I want to mention a big limitation of that study that I just talked to you about. And that is that everything that we did there was taking all genes at the same time and their co-expression to predict whole brain functional connectivity. But if we actually study the correlation coefficient between the gene co-expression matrix using all genes and functional connectivity, that correlation value is about 0.1 to 0.2. So not huge. We're not predicting a lot of variance. And if you go back to the slide where I had all of the different frequency bands, you just looked at that tiny gene co-expression bar, it was very small, right? So that's not necessarily doing a great job in explaining functional connectivity. However, if instead you allow one set of genes to code for one particular node's connectivity or one edge, and another set of genes to code for another edge or another node in the network, then you can actually get much higher. And in fact, what we have done in this paper from Max Berlero that's on arXiv as a preprint, we've asked, are there sets of 50 to 200 genes that together account for functional connectivity of a single brain region? And is it a different set for different brain regions? By doing that and allowing that variability across genes to predict patterns of functional connectivity, we can get the correlation much higher, and we can account for now up to 40% of the variance in functional connectivity. So that's a pretty drastic change, and perhaps is more relevant to the biology. So here is the idea, which is that there may be sets of genes down here that code for the connectivity of sets of brain regions, basically allowing one gene module to explain variance in one brain module. And so this is the picture of sort of what we were hypothesizing to be the case. And then here is the data. The color code here and the color code of this bar is the modular structure of resting state bold, which is shown here on the surface. And what you're seeing in here is the patterns of gene co-expression that have now been just taken from the gene set that is most predictive of that module's connectivity. So you can see a really nice modular structure here, which recapitulates the modular structure of the resting state bold. And I think that's suggesting that the functional connectivity that we are observing is modularly encoded in the genome, and it's not the case that the same set of genes is relevant for every region in the brain or every module. And so I think with that knowledge in mind, it's useful to go back to these suggestions for future work that I just described to you and say, is it possible that when we build these models, we should be allowing for distinct sets of genes to differentially contribute to the prediction at different brain regions? And I think that that would probably allow us to do a better job in prediction, but also be more true to the underlying biology. Okay, now with that, I'd like to go on to the question of informing mechanistic models. And here I am motivated by the question of, you know, it's all fine and good to study structure and gene co-expression and functional connectivity, but humans, what we care about in humans often is their behavior. So what we'd like to do is to use multimodal imaging not just to understand the basic biology, but also to understand behavior. The particular kind of behavior that I'm interested in this study is working memory. And there's actually some very nice prior work that's demonstrated from several different labs and different data samples that the strength of functional interactions between the frontal parietal system and the default mode system during an NBAC working memory task is correlated with working memory performance. In what I'm going to show you in a couple minutes, I will be taking bold fMRI data that's acquired while somebody is performing the NBAC task. I'll be estimating the functional connectivity between two regions using a Pearson correlation, although all of the results also hold if you use a wavelet-based coherence instead. And then here, I'm showing you the pattern of functional connectivity across all regions and then collapsed to individual systems. So that's where I take the average inside any one of these blocks, meaning the regions that are associated with a particular system. Okay, so we're studying specifically the Human Connectome project data that's been publicly released from the S1200 release. And here, what we find, consistent with prior studies, is that the strength of the functional connectivity between the frontal parietal system and the default mode system during an NBAC task is inversely correlated with accuracy. So here is the strength of the frontal parietal default mode connection, and then here's two-back performance. This is accuracy, although the same effects hold if you study D prime instead as your behavioral measure. And then here's that same strength of the FP-DM connection versus zero-back performance, and you can see that there is not a significant relationship here. So this seems to be a functional relationship between two systems that's very specific to two-back working memory performance. On the right-hand side, I'm showing you the areas on the cortical surface that we're assigning to the frontal parietal system in orange, and then the default mode system in red. And those colors will stay the same for the next few minutes, so that you know. So our aim here was to use multimodal neural phenotypes, including regional activity, interregional connectivity, and structural linkages to explain individual differences in working memory. And I'd like to mention here, again, a big limitation of prior work, but also what I've shown you so far, which is just that functional connectivity, as we know, is completely blind to activation. And so while we often have intuitions about a task and about cognitive processes from activation studies using general linear models, for example, we often don't necessarily know how to translate those intuitions to patterns of functional connectivity or vice versa. And so I think there is a big open space where we need to start linking together how to understand changes in patterns of activation and changes in functional connectivity. So here what I'm showing you is we perform a general linear model to estimate beta weights of the 2-back minus the 0-back contrast. And then we average those beta weights over the default mode system. That's what's in the y-axis here. And then we also average them over the frontal parietal system, and that's what's shown here. So what you're seeing is the pattern of activity in the default mode system is positively correlated with frontal parietal default mode connectivity. And then the activity in the frontal parietal system is negatively correlated with frontal parietal default mode connectivity. So that's interesting. It suggests that activation in these two systems is differentially related to patterns of functional connectivity between them. Each data point here is a different individual in the Human Connectome project data set. Now, the next question we had was, which of these is possibly more or less related to end-back working memory performance? And what we found is that the beta weights specifically in the frontal parietal system are correlated with performance, but the beta weights inside the default mode system are not correlated with performance. So that suggests that there may be a specific role for the frontal parietal system and its activity level in supporting working memory function. Okay, so I want to ask, what is going on here? Can we have a hypothesis about how activation and functional connectivity work together in these two systems to support working memory performance? So we proposed that the dynamics of this frontal parietal system directly modulates the strength of the functional connection between the default mode and the frontal parietal system. So here is just a schematic to illustrate what I'm going to try to explain, which is that here's the time series in the default mode system, and you can see it oscillating here. And then here is the frontal parietal system in blue. And then what I'm showing you in orange and green is the notion that there could be two separate parts of the frontal parietal system that are differentially connected to the default mode. So our candidate mechanism assumes that this frontal parietal system is composed of two systems. I'll call them the orange and the green. They're non-overlapping spatially. And we suggest that they are separately tuning the strength of the connectivity between the frontal parietal system and the default mode system, such that when one of these networks is more active, that tunes the connectivity one way, and when the other subnetwork is active, it tunes the connectivity the other way. They're sort of competitive with one another. And this is based on some prior work suggesting that the frontal parietal system is divisible into two different subnetworks, and also by the observation that this is a relatively parsimonious way to explain the patterns that we're observing so far. All right, so to test that hypothesis, the first thing that we need to do is to separate the frontal parietal system into two sections. So we use a data-driven approach, and specifically we're using weighted stochastic block models, and I'll just make a note for people who are interested in the methods that we're becoming more and more excited about the weighted stochastic block model as a method for community detection. And largely that's because, unlike modularity maximization, it is able to identify or separate the network into groups that are not necessarily strongly internally dense the way modularity maximization would do, but it can separate and identify these dense modules, sort of less dense modules, and anything in between. It can also be sensitive to core periphery structure and to bipartite structure, so it's a much more general model that I think is sensitive to greater variation in the patterns of functional connectivity that we observe in the human brain. So anyway, when we use the weighted stochastic block model on just the frontal parietal network connectivity, we use a maximum likelihood to identify what the optimal number should be of subnetworks. We identified two. We're going to call them subnetwork A and subnetwork B. They're shown here in orange and green. And then what we'd like to do is to ask, well, they are separable from functional connectivity, but are they also separable in other ways? So here's where the multimodal data comes in. So what we did is that in these same individuals, we studied their structural connectivity from diffusion imaging, and we looked at subnetwork A and subnetwork B, and we found that they are differentially structurally connected to the default mode system. So we find one network being more strongly connected to the default mode system, that's this green one here, and the other subnetwork, orange, being more structurally connected to the dorsal attention system. So I'll just walk you through this. On the y-axis is strength of structural connection between each subnetwork and the default mode. Here you see that subnetwork A preferentially connects to the default mode system. And then here you see on the second graph, strength of structural connectivity between that subnetwork and the dorsal attention system. And here you see that it's subnetwork B that is more strongly structurally connected to the dorsal attention system. So this suggests to us that these two subnetworks are not just functionally distinct, which we can observe from the weighted stochastic block model, but also structurally distinct, suggesting they're possibly able to differentially impact system-wide dynamics, or there's a structural support for that claim. So then the second question we wanted to address was whether these two subnetworks are also genetically distinct, just as another piece of information to say that the separation of the frontal parietal system into two sections is important and biologically relevant. And so to address that question, we first quantified the average magnitude of gene co-expression for pairs of regions that are located in a single subnetwork. So basically, we take all regions that are in the subnetwork A and calculate the gene co-expression between them, and then separately all of the regions that are in subnetwork B. What's the average co-expression between them? And then we quantify the average magnitude for all of the pairs of regions between the two subnetworks. So if I take one region in subnetwork A and one region in subnetwork B, what is the what is the average gene co-expression for those two? And what we consistently observe is that the gene co-expression for regions inside of subnetwork A is significantly higher than the expression for two regions that are between subnetwork A and B. And we find the same basic point for subnetwork B, which is that the co-expression inside subnetwork B is significantly higher than gene co-expression between two regions that are located in the two different subnetworks. So that's now three pieces of information supporting the notion that the frontal parietal system can be separated into two different subnetworks that are functionally distinct from end-back working memory task bowl data. They're structurally distinct from diffusion imaging data, and they're genetically distinct using the expression data from the Allen Brain Institute. Okay, so then now that we've identified that we can separate the frontal parietal system into these two sections, can we go back to our hypothesis and ask whether those two subnetworks are differentially tracking functional connectivity with the default mode? So here what you see is the strength of the connection between each subnetwork and the default mode. And this is a functional connection. And what you observe is that one subnetwork is positively connected with the default mode system, and the other one is negatively connected. Now remember, this subnetwork A was also the one that was structurally more linked to the default mode, right? And now it's positively connected with the default mode functionally. And then more importantly, however, is this piece. Here on the y-axis is the strength of the frontal parietal default mode connection, and this is the activation of subnetwork A. And what you can see is that when subnetwork A is more active, the frontal parietal default mode connection strengthens, and then the inverse is true about subnetwork B. So when subnetwork B becomes more active, the strength of the frontal parietal system, of this interaction between the frontal parietal and default mode system decreases. What that suggests is that these two systems, when they change in their activity, it is altering the strength of functional connectivity between these two large-scale areas. Okay, but that's still just correlative evidence. What we'd like to do is to build a model that tests that mechanism in silico using a computational approach. So what we did is that we built a simulation where we had the default mode system, the subnetwork A of the frontal parietal, subnetwork B of the frontal parietal, and we also included the dorsal attention because if you remember when we looked at the structural connectivity, here we actually found that subnetwork B was more strongly related to the dorsal attention and subnetwork A was not. So this effect seems to also potentially be important. So we included the dorsal attention system in the model as well. There's also additional data from previous papers and previous labs that supports the suggestion that there may be a role for the dorsal attention system in here too. Okay, so then we instantiated a Stewart Landau model, which allows us basically to study the normal form of a supercritical Hopf bifurcation. We use or inform that model with structural connectivity between the systems. So the actual structural connections between each of these different systems have a weight that's the coupling is given by the strength of the diffusion tractography edge that we estimated from healthy individuals. And also the natural frequencies of the oscillators are taken from the bold data, resting state bold data. Lastly, the network dynamics were integrated out for a total time of 6 minutes, which is consistent with the length of the empirical end-back scan, so that we were measuring sort of the same amount of data. And here we began by just starting simple, where each system was its own oscillator, which means there are only 4 oscillators in this simplified model. And what you see here is when we increase the activity of subnetwork A, leaving everything else the same, not touching anything else, just increasing the activity of subnetwork A, we see that the frontal parietal default mode connection strengthens. And then here, when we increase the activity of subnetwork B in the model, but again leave everything else alone, then we decrease the strength of the functional connection between frontal parietal system and default mode system, consistent with the data. So that's suggestive that just increasing the activity of each of these subnetworks differentially tunes the functional connectivity between the two systems. But 4 units is a very, very simple model. One other step of sophistication is to say, well, we know that each of these systems has a different number of regions inside of it. And so we can actually use the Schaefer atlas to say how many units should be in each region. So now the default mode system will have a certain number of units, and that number of units is equal to the number of regions in the default mode system in the Schaefer postulation, and the same for the rest of them. So this gives us a model with 192 units. And again, if you increase subnetwork A activity, you get the strengthening of the functional connection. If you increase subnetwork B's activity, you get a decrease in the strength of this functional connection. So consistent results. But lastly, we also know that these systems don't act independently from the rest of the brain. They're situated inside of many other systems that also have their own connections. And so we can actually use just the full Schaefer atlas, so a full 400 units. Every region is now an oscillator, and the structural connection between each pair of regions is the edge in the diffusion-weighted network. And again, we see the same pattern, where increasing subnetwork A activity strengthens the connection, and increasing subnetwork B activity decreases the connection. So that's suggesting, again, that using a computational model, we can ask whether the change in activity of these specific subnetworks is a sufficient explanation for the observed change in functional connectivity, supporting working memory performance. All right, so just to summarize this study, We started by recalling that individual differences in frontal parietal activity, but also in frontal parietal default mode connectivity track individual differences in working memory performance, both in accuracy and in D prime. We posited that the dynamics of the frontal parietal system directly modulate the strength of the functional connections between the frontal parietal and default mode system via a two-set subnetwork structure. And then subnetworks, we went and looked for them. We detected them in a data-driven fashion using the weighted stochastic block model. We found that they were also distinguishable in terms of structure and distinguishable in terms of gene co-expression. And then lastly, we built a multimodal data, built a biophysical model using multimodal data to test that mechanism and found consistent findings suggesting that changes in activation can drive changes in functional connectivity, supporting working memory performance. Okay, so that brings me to just sort of the last big picture, which is why and how multimodal neuroimaging. As we talked about at the very beginning, number one, to explain observations in modality late in the causal chain, like functional connectivity, from observations earlier in the causal chain, like structure. Number two, to build generative models that can bolster inferences from sparsely sampled patient data. And number three, to inform mechanistic models. With that, I'd like to acknowledge the people who were important in the work. In particular, the first study was led by Rick Betzel, who's now a professor at Indiana University. The second study, that the functional connectivity is modularly encoded in the genome, was led by Max Bertolero, who's a postdoc in the lab now. And the last study on working memory was led by Andrew Murphy, and I don't have a picture for him, so he's a Rainbow Network instead. All right, thanks. If anybody has questions, I'm happy to answer.
We have time for about one question.
Thank you very much for this talk. I was wondering about the differences in the electrocorticography where the higher frequencies predict more of the connectivity.
Yeah.
Now, there has been some evidence in the EEG world that some frequencies are related to connectivity between short-range connectivity versus long-range connectivity for higher frequencies, if I'm not mistaken.
Yeah.
Do you think that this has anything to do with that? And related to that question is something that came later in the talk. I kind of lost my train of thought.
Okay, so for the first part then, yes, I think that, so there is evidence that the high frequency signals are relatively show short range coupling rather than long range coupling. And so it's possible that that short range coupling is very well predicted by physical distance, which is one of the predictors in our model, but it's also very strongly related to the search information, which was one of the other predictors in our model. And so I think the fact that we have two different pieces of information that are both sensitive to that local coupling effect indicates or is an explanation for why we're doing better in the high frequencies. That suggests that there's something else going on at the low frequencies that require, I think we need to think a little bit harder about whether we should be including a different predictor inside of the model. I will mention that in separate work, we're also asking the question of, the search information is just one measurement of communication on a structural network, and there are many others that can be sensitive to local coupling versus distant coupling. And so we can actually separately test all of them, or a reasonable set of them, and determine which one is better predictive at the low frequencies.
So in the computational part, you talked about an oscillator being assigned to each cortical region. So what was the frequency of that oscillator, or how was it modeled in terms of oscillative properties?
Yeah, the natural frequency was chosen from that region's resting state bold data, so it was data-driven, which is also why we're simulating out to the 6 minutes. So it's very slow.
not based on the EEG data. That would be more pertinent in a way.
Yeah, the second half of the talk was did not use ECOG data.
Okay.
Yeah.
Thank you.
I think it would be interesting to use that, but we didn't.
Yeah, let's thank our speaker one more time.
Thank you.